Okay, welcome to Primate Pro, and today we will continue from cilia and flagella, and we will try to complete the syllabus by today. Later then in the next day I will give a gist video about the whole chapter, which will be very much beneficial during the exam days. Okay, before exam you need to study on a single lecture and to review the any concepts, and because this 8 to 9 lectures will not be able to be viewed before the exams, because at that time large number of studies will be overwhelmed. So I will try to give a concise video lecture of the total chapter later in these videos. Okay, now we will start from the cilia and the flagella. Cilia and flagella. Cilia and flagella, these arise from centriole. Like structure called basal body. Okay. Now centrosome and centriole. Centrosome and centriole. Centrosome consists of two cylindrical structure. Centriole this one, the angle between, okay, let me draw it in a good manner. The angle between this is generally 90 degree, generally perpendicular. Okay, generally it is perpendicular. Okay, now these are the proteinaceous region that is being surrounded. These are proteinaceous region. Okay, now it is present in cytoplasm. And cytoplasm is present in animal cell, that is animal cell. Absent in almost plant. Okay, it is absent in almost plants. Okay, it is absent in almost any plants. Now let me draw a structure. Okay, so here CBA, I will later index it. CBA, A, B, C. A, B, C, A, B, C. Okay, these are the these are the triplets. These are called triplets, and these AC linker. Now you see the periphery of the 9 triplets. The periphery of the 9 triplets. This can be said in notes. Periphery 9 triplets. Okay, one triplet, 3 emoji A, B and C consist of one triplet. So total peripheral microtubule that is MT equals to 9 triplet is equal to 9 into 3 is equal to 27 MT. Okay, now center no microtubule but proteinaceous structure. No microtubule but the proteinaceous structure, that is called HUBB, hub and spoke like model. Okay, no proteinaceous structure is there. Now radial spoke 9 connect to A and of microtubule. Two peripheral triplet connected by AC linker. Okay, two peripheral triplet is connected by AC linker. Now cartwheel structure. This wheel is called the cartwheel structure because here everything is connected. C to A. So it's a cartwheel structure. Okay, cartwheel structure. 9 triplet plus 1 center. It's periphery. Note: centrosome, 2 centriole is 27 plus 27 equals to 54 MT microtubule. MT is microtubule. Okay. Note: cilia and flagella 9 doublet plus 2. Now centriole or the basal body. See, the centriole and the basal body are of 9 triplet. So cilia and flagella are doublet and centriole and the basal body is triplet. 9 triplet plus 0. So cilia and flagella, this question comes, cilia and flagella called as 9 plus 2 structure. Okay. Centriole forms basal body of cilia or flagella and spindle fiber animal cell. Okay, done. Now we comes to as nucleus. We come to nucleus. Give the topic nucleus. Nucleus. Discovery by Robert Brown in the year 1831. Okay. Now generally single nucleus per cell. Note paramecium. Two nucleus, mature mammalian, RBC, erythrocyte, and sieve tube of phloem. Nucleus is absent. Structure. A nuclear envelope / nuclear membrane, NM. Chromatin, nucleus and nucleoplasm / nuclear matrix. Nucleoplasm / nuclear matrix. Now we come to nuclear envelope. न्यूक्लियर एनवेलप क्या होता है? न्यूक्लियर एनवेलप, न्यूक्लियस जिसके अंदर रहता है उसको ही न्यूक्लियर एनवेलप कहा जाता है. तो न्यूक्लियर एनवेलप हेडिंग डाल दीजिए, न्यूक्लियर एनवेलप. न्यूक्लियर एनवेलप, now I have to draw a structure. Okay. न्यूक्लियर एनवेलप, न्यूक्लियर एनवेलप क्या होता है? This is Now I am drawing here rough endoplasmic reticulum, that is, it contains ribosome. Okay, this is RER, and outer membrane, that is inner membrane. This is inner membrane, continuous with, outer membrane is continuous with RER. राइबोसोम इज प्रेजेंट, इधर पे राइबोसोम प्रेजेंट है और इनर मेम्ब्रेन में राइबोसोम एब्सेंट है, ओके. नाउ पेरीन्यूक्लियर स्पेस, दिस इज कॉल्ड द पेरीन्यूक्लियर स्पेस, present between outer nuclear membrane and inner nuclear membrane. It is 10 to 50 nanometer. Note, nuclear membrane oblique perinuclear space separate cytoplasm and nucleoplasm End of nuclear membrane fuse, so a space is created. It is nuclear pore. Nuclear lamina, nuclear lamina is proteinaceous, present on inner nuclear membrane. And nucleopore, that is the nucleopore, this nucleopore, it controls bidirectional movement of RNA and protein, controls bidirectional movement of RNA and protein. ओके, आरएनए और प्रोटीन का जो बाइडायरेक्शनल मूवमेंट है वो न्यूक्लियोपोर के द्वारा कंट्रोल किया जाता है, ठीक है ना. उसके बाद हम आएंगे, गिव द हेडिंग क्रोमैटिन. क्रोमैटिन और क्रोमोसोम के डिफरेंस से ज्यादा सवाल आता है. Questions come in the NEET from the difference between the chromatin and the chromosome. So I will write a differentiating a table between the chromatin and the chromosome. I am drawing the table. Okay, this is chromatin. Who discovered it? Named by Fleming. Alexander Fleming. And this is chromosome. Okay, it is long and thin, loosely packed, less folded DNA, least condensed, less visibility, it is more complex, indistinct, not clearly visible, interphase, cell non-dividing. This is short and thick, tightly packed, more folded and more condensed, more visible. So chromosome is more condensed and more visible. Okay, now it is distinct and dividing stage, prophase, metaphase. Okay, now we come to, after that we are coming to give the heading as packaging of DNA. Now DNA is negatively charged, bind with histone protein, histone protein are basic in charge, basic in pH is basic, positively charged, amino acid, arginine and lysine, basically arginine and lysine, H2A, H2B, H3 and H4 all consist of 2, 2, 2 and 2 units, making up octamer of histone protein plus the DNA. Okay, this consists of packaging of DNA. Okay, now the last is the chromosome. Very less will be required in this chapter. Chromosome, structure. This only comes when the centromere, this is the centromere, is at center, it is say metacentric. This is metacentric. when the centromere moves towards end, making a long tail, short tail, or short arm, basically short arm. This will not be a tail, this will be arm. This is submetacentric. When it moves more towards other end, it is acrocentric. Sorry, this is telocentric. When it goes to end, it is acrocentric. Okay, here we can conclude this chapter. No more questions will be asked excluding these notes, but I will give you a gist of this chapter in the next video. The total chapter will be concised in a single video of 30 to 35 minutes, which will be very much beneficial for you guys before the exam. These are the tips. Okay, thank you, and I wish to see you again on the next video. Thank you.